Yeah, that's what I was thinking.
All right, we're gonna go ahead and get started. Sorry, we're running a couple minutes late. We just wanted to get office hours set up for everyone because I know the exam's coming up this week, so wanted to make sure there was maximum amount of availability for you guys to come and ask your questions. So first off, there is the review session tonight at 7:30 p.m. It's in this room. If you can't make it, it will also be recorded and posted. I know it's a little last minute, came together in a short time frame, so future exams, it should come together a little earlier and hopefully give you a little more warning for that. And then the hypothesis assignment is also due tonight. So if you have any questions about that, feel free to reach out, but don't feel like you need to put too much pressure. These assignments are for you so that we can give you feedback to make sure you're on the right track. So you don't need to worry about getting a right grade. This is not factored into your final grade. So this is mostly just to help make sure you're developing your project in the right direction so that we can help you early with these things. And then finally, office hours for the next 2 days. I just wanted to lay all of those times out because a couple of them are shifted around. So for tomorrow, Wednesday, I will have office hours from 4 to 5. Shri will have office hours from 5 to 6. Nejra will have office hours from 10 to 11 in the morning. And Dan's will be from 9 to 10 in the morning tomorrow. So plenty of time in the morning and in the evening. And then on Thursday, both Ahava and Cooper will have office hours before the exam. So Ahava's are 10 to 11 and Cooper's are from 12:30 to 1:30. And if you need locations on any of those, check out the Canvas or email any of us just so that you have enough time to go through any questions you have about old quizzes or any content related material, or if you just want to come talk through the material with somebody to make sure you're understanding it well. We want you guys to crush it on the exam. So I know you're going to do great. Don't worry too much. But for now, we're going to jump into the last lecture before that. So this is our second lecture on molecular motors. So last time we talked about tubulin-based motors, the kinesins and dyneins, and today we're going to talk about actin-based motors, and these are generally the myosin family motors. So we're going to talk a little bit about cell movement, but mostly about the sarcomere that is found in striated muscle, and as an example for how these actin motors work and are moving, and specifically in terms of the cross-bridge cycle. So we'll get into fine detail about all that, but as always, feel free to interrupt me at any time. Even if I'm going too fast, just tell me to slow down. Yes. No, tomorrow from 6 to 7, Shri is having her office hours. Yeah, I might have forgotten to say that one. Thank you for clarifying. Yes.
About the quiz on Thursday. Are we still going to go over like the quiz answers right after the quiz?
Yes, yeah. So we'll have the quiz. We'll go over the answers right after, and then after we cover any questions about that quiz, we will start the exam. So we won't do like we did last week and discuss questions at the end. We'll just take out all the questions right there so you have all the information before you start the exam. Good question. Are there any other logistical questions before the exam about the assignment? Nope. Okay, great. All right, so our disease topic for today that we'll be using to demonstrate is heart failure. So we'll go ahead and get into that. Heart failure is defined as when a person's heart can't pump enough to meet the need for the body. So sometimes this is thought of in terms of ejection fraction of the heart. This is one of the clinical measurements, and this can be due to a number of reasons, but ultimately leads to less oxygen and cardiac contractility. So some of the symptoms of this that might be familiar, dyspnea, we talked about already. Does anyone remember what that is? This is, yes. Yeah, difficulty breathing, so shortness of breath. Wheezing is similar in that case. Edema is a swelling or a local buildup of fluid. Fatigue and nausea, impaired thinking, increased heart rate, all of these come about when your body is not getting the oxygen that it needs. So when you're not pumping enough blood throughout the body. And heart failure can be caused by a large variety of underlying conditions. So coronary artery disease is a big one. This is when there's plaque buildup in your blood vessels or on your heart. Myocardial infarction, so also known as MI, this is the word for heart attack. So this happens when there's some kind of blood circulation cut off to your heart, and then that tissue begins to die. But then once circulation is restored, the tissue can't regenerate itself. So heart is a really poor tissue at regenerating itself. And so there's some kind of scar buildup there. Similar to what we talked about with fibrosis, this happens in heart attack as well. Hypertension is another one. Does anyone want to define hypertension? Yeah. High blood pressure, exactly. Congenital heart disease is another potential underlying cause of heart failure. And so congenital just meaning something that is from birth. So this is like a birth defect in the heart anatomy. Diabetes is another one. Hypertrophic cardiomyopathy is one too. This is just the increased growth of the cardiomyocytes or the muscle cells that are in your heart, leading to an enlargement of the heart and differences in the biomechanics of the blood pumping. Okay, so heart failure is a really important disease topic to touch on because it is the leading cause of death in the US, and partly because there's so many different underlying diseases that can lead to it, and your heart is such a vital organ. So some of these statistics are fairly old, but 5.1 million people in the US suffer from heart failure. This is, of course, a huge number, and 1 in 9 deaths back in 2009. So this is a truly devastating disease, and there's not great prognosis. So in terms of treatments, usually about 50% of the people who suffer from heart failure end up in morbidity after 5 years. And because of this huge population, the costs of this disease are in the billions. So kind of to put into perspective, the NIH, the National Institute of Health, that funds all medical research, this is almost their entire budget for funding all medical research in the United States. So this is a hugely devastating disease for a number of reasons. And interestingly, there's differences in presentation geographically for this disease. So this figure was taken from the CDC, and the darker colors are higher prevalence of heart failure. And you can see kind of regionally, especially in these southern regions of the United States, there is kind of a localization of increased incidence of heart failure. And this could be potentially tied to a number of causes, maybe differences in socioeconomic status or perceptions of mobility or other things like this, but it's not exactly known. It's just an interesting effect of or presentation of heart failure. All right. So, like I said, we're going to be talking about myosin motors today. So these are the actin-based motors. And similar to kinesins, there is a lot of types. So this is a family or a superfamily of molecular motors. And the one that we think of most generally and that we're going to talk about extensively today is myosin 2, which can also be called muscle myosin or conventional myosin. And this is thought of a lot because of its large number in the body. All of your smooth muscle, or sorry, all of your striated muscle is composed of myosin 2, and this is any muscle that you're using in your skeletal system or in your heart. So another reason why we'll focus on this today, but there are also many other types of myosins, and they are for a large number of purposes, either for cargo, transport, like we talked with a lot of the kinesins and the dyneins, or for other types of contractility. sorry. We'll keep going. We'll talk about myosin 2 and 5 later. So with the few exceptions in the cargo transport and also in the contraction-based myosin activity, these are often plus end directed. So they're moving along the plus end of the actin filaments. And similar to kinesins and dyneins, they have these motor domain heads and tails that are binding different things. In terms of muscle, the tails bind together and form these filament-like structures. So that's what you're seeing in this example here on the right, is a number of these myosin heads, and then their tails are kind of congregating in the middle to form this filament. Myosin 1 and 5, like I said, these can be cargo binding. So this is an example. It's a little bit of a strange picture, but these can have 1 or 2 motor head domains and be binding cargo on their tail domains. So this yellow slab here is kind of representing just like a generic cargo that they might be binding. Okay, so in terms of regulation, there's 2 general types of regulation that we'll be talking about, coming back to a bunch in this lecture. And one of them is blocking the binding sites on actin. So this is more of an actin-centric regulation approach. And this is occurring a lot in striated muscle, like I said, so this skeletal and cardiac muscle. And then the other approach is more of a myosin-centric approach. So this is binding the motorhead or blocking the motorhead activity in some form or fashion. And this is happening in everything else. So in your smooth muscle, as well as in the cargo transport activity of myosins. All right, so we're going to talk a little bit about the structure. So what we see here is the sarcomere, and this is the functional unit that makes up striated muscle. So if you see here on the left is an electron micrograph of kind of a cross section of striated muscle, and you can see these rectangular units. So each of these rectangular units would be 1 sarcomere. And you can see it kind of looks like there's stripes going up and down and across. So that's what striated means, similar where this word comes from. So a little bit about the structure. We're going to zoom in onto one of these sarcomeres and talk about this extensively. So starting kind of on the ends here, we have these 2 protein structures known as the Z band. And these Z bands are binding the actin filaments. So this is the cytoskeleton component of actin, and they're binding it at the plus end. So the actin is oriented so that the plus ends are both outward and are connected to the Z band. And then on the inside here, we have the same thing that we were showing on the other side. We have these bundles or fibers of our myosin 2 that are coming together to form this filament. So we talk about the actin filament and the myosin filament as our thin and our thick filament. And this is just a function of the diameter of these. So if you remember our discussion about the cytoskeleton, the actin is fairly small in diameter, only about 5 to 7 nanometers. And the aggregation of all of these different myosin motors coming together forms a filament that's larger than actin. So that's why we call it the thick filament compared to the thin filament of actin. And then where they bind here, oh, I lost my, where they bind here together is what we call the cross bridge. So this is just the connection point between these thin and thick filaments. And this is where the activity of the contraction or the molecular motor component of this is being facilitated. Yes. You could say that it's a little hard if you're looking at this to see the actual difference actin and myosin. I was thinking out of it of more of a zoomed out view. So the organization of the sarcomeres looks like stripes. But in terms of what is striated muscle, these kind of sarcomere units are only existing in striated muscle. Does that help a little bit? Okay. So the thing in the center is called the M band. This isn't one of the blue terms, but there's a lot of different components. It kind of binds together the other protein units. So these are not in this description, but this spring-like structure here, this is another protein called titin, and it's binding also to the Z band, and it's moving all the way into the middle and binding this M band in the middle. So it's kind of a structural component. This is a really big protein. It's actually the biggest protein in the body. And it's very elastic, so it can stretch as the contraction moves. But again, these aren't blue terms. This is a little more detailed than we'll need for this demonstration, but good question. Yes. Yeah, good question. So this schematic here on the right is composing one sarcomere. So yeah, in this picture on the left here, these rectangular units, like this would be one sarcomere, this would be one sarcomere, etc. Yeah. Yes. Yeah, so the thick filament is just these bundles of myosin motors. So their tail ends kind of wrap around each other and creates this filament with the motor head domain sticking out. So just that component is considered the thick filament, where that connects to the thin filament is what's known as the cross bridge. Great. All right, we're gonna zoom in a little bit more. So there's different regulatory components that, oh, let me make sure I didn't. Sorry, there's a blue term that I missed on this slide. Let me just make sure I cover all these bullet points. So we talked about the structure a little bit, but in terms of the regulation, this is one of those actin-centric regulations in terms of the sarcomeres. So this is going to be regulated in terms of the availability of binding to actin. So specifically blocking these sites where these cross bridges will form. We'll talk about that more in detail. But this inhibition is driven or relieved by calcium availability. So when there is an abundance of calcium, this is known as a disinhibition. It's a little bit of a double negative, but the abundance of calcium allows these cross bridges to form and these sarcomeres to contract. And we'll talk about that, why that is in the next slides. But just want to make sure I define that blue term. And again, this is based in the thin filament, so actin-centric. Okay. Continue on. So some of these regulatory components are shown in this schematic here. So there's a lot going on. We'll first look at the actin component. So we have these different protein structures. We have this filament known as tropomyosin, and this is wrapping around the actin filament. So this is a coiled, two-stranded filamental protein that's closely associated with the actin cytoskeleton. And it has this functional unit here that is kind of binding it to actin. So this is composed of troponin T, troponin I, troponin C. We'll talk about what all those components do on the next slide. But then a little bit later, we'll talk more about these myosin-centric regulatory elements. So this regulatory light chain in particular that's binding on the thick filament. So we'll come back to this later, but just in terms of what it looks like to have actin-centric or myosin-centric regulation. Okay, so this troponin complex, like I mentioned, has 3 different protein subunits, and they're acting in different ways. So the troponin T is binding tropomyosin. So this is this functional glue, the side that's connected to tropomyosin, whereas the troponin I is binding the actin. So it's kind of the other side of the glue connecting this unit to actin. It's overall bridging actin to tropomyosin, keeping them associated with each other. This troponin C is kind of the functional unit that's doing the regulation. So this is the unit that is going to be responding to calcium, binding it, and changing its conformation that is enabling the exposure or blocking of the actin binding sites. So this is where the activity between the myosin head motor domains and the actin filament are occurring. So the naming structure is kind of nice here. TIC kind of gives you a clue as to what it might be doing. Okay, and a lot of these cartoon diagrams are helpful in terms of how you can think about the function of these protein subunits. But this is a cryo-EM structure that shows what this actually looks like. So this doesn't show the troponin complex, but this does show the actin filament and what this tropomyosin looks like closely associated to it. So the tropomyosin is here in yellow, and then these are just the different actin subunits. So this little red here is the ADP, so kind of a reminder back to our cytoskeleton lecture where these actin monomers are ATPases. So it's still going to be binding the hydrolyzed ATP in the form of ADP after it's polymerized. Okay, so how does this regulation actually work? There's a couple different theories. There's kind of a classical theory and more of an updated theory in terms of how the availability or blocking of these actin binding sites occurs. So like I mentioned before, there's general consensus that this is caused by a conformational change in the C component of this troponin complex. But the classic theory is just that there is some kind of opening and closing. So this moves when the troponin C is binds to calcium, and then it moves back when it's not. So what this kind of looks like, this animation kind of demonstrates it. So the red line here would be the actin, or the tropomyosin, sorry, and the blue is the actin filament, where these smaller blue circles would be the binding sites for the myosin heads. And so when calcium comes in, it's going to bind to that troponin C part of the complex. It's going to change conformation, and then it's going to shift this whole tropomyosin out of the way and allow the binding sites for the myosin motor heads, which makes perfect sense. This is kind of a simple and intuitive way that you can think about this happening. But there's been some more recent evidence. As we've said before, this is kind of a class of half-truths, so things are always changing. There's always new developments in the field. And kind of an updated explanation of this is a little more nuanced, but fits in with some of the concepts that we've talked about before. And so this would be that this troponin I is more so working to lock the tropomyosin in place. So when this troponin I is not bound, then the tropomyosin is free to move, to diffuse around, and it can be pushed out of the way by the myosin motor heads, able to bind the actin filament. But when there's no calcium around, it changes the conformation of troponin C that continues to then lock the tropomyosin in place and block these actin binding units. Does that make sense? Is there any questions? I know it's kind of a subtle difference between these two, but it brings the troponin I a little bit more into play, as well as the Brownian movement, or the random movement of things that are happening on this nanoscale level. Great. That's clear. We'll keep going. If you have any other questions about it, feel free to bring it up at any time. Okay, so this is kind of a summary again about how this pathway works. So there's some trigger that causes the intracellular stores of calcium to be released into the cytoplasm, and that abundance of calcium will bind to troponin C, and then that troponin C will change its conformation, allosterically regulating troponin I, and that troponin I will then be released from it. It's locking the tropomyosin into place, and that allows the movement of tropomyosin and the accessibility of these myosin binding sites on actin. So a couple steps here, but a few main players just to understand and be aware of. All right, so now zooming in a little more, now that the actin binding sites are available for myosin to come in and associate, now they can actually bind and facilitate this movement. So the binding and movement of these motor proteins is often referred to the actin-myosin cross-bridge cycle. So there's a lot going on here. There's a bunch of steps. We're gonna walk through it slowly, but a couple things to keep in mind here. So one thing I want to point out is just the polarity of the actin filaments in this schematic. That kind of helps orient which way the motors are moving and the purpose of these conformational changes that we'll talk about. So the myosin motors are moving along actin in the plus-ended direction. So this, in the schematic, these would be moving to the left. And these myosin motor heads are also enzymes. So these kind of, just like actin, are ATPases, meaning they bind ATP and they hydrolyze one of the phosphates, and that creates ADP as a result. So ADP plus inorganic phosphate. So the ATP and ADP bound myosin are going to have different conformations. So, sorry, another thing I'll point out about this schematic is the words ADP here, or ATP or ADP-P. This is just referring to what is actually currently bound to this myosin motor head. So as we step through these, this is like, you can imagine this as the molecule bound to the motor head. So because these myosin motor heads are enzymes, they're going to have differential affinities for their substrate or the product. So generally, you can think that enzymes are going to have a high affinity for their substrate and a lower affinity for their product. So they'll want to bind their substrate, but then once the catalytic reaction occurs, then they'll want to release that product into the cytoplasm. But something interesting that's going on here is, as this enzyme, as this motor head is changing its conformation in this enzymatic reaction, it's also changing the affinity that myosin has to bind to actin. So the red here in this schematic is referring to a strong binding interaction between myosin and the actin filament, not necessarily the affinity between the myosin and its substrate at that point. So generally, myosin is going to have a strong affinity for ATP and a lower affinity for ADP. Okay, does that all make sense before we walk through? I think it helps to kind of get the lay of the land of the schematic. So because this is a cycle, we could really start anywhere, but we'll start right here on the bottom right, in which the motor head is kind of cocked back. It's in this perpendicular orientation, and it's bound to ADP. So this is its product in this case. So when it's bound to ADP and it's in this conformation, it has a strong affinity for actin. So it's binding it, and this is known as the Rigor state. So this comes from just different studies that were done on corpses when people were learning about anatomy. So when somebody dies, their cells are no longer working or active. There's not a production of ATP, and so there's predominantly ADP available. And so this is the state that these myosin motors will be in all of the muscle of tissues that were being investigated. So this rigor state kind of refers to this state of rigor mortis, which is when somebody dies, if their muscles will tense up like that. And that's because the myosin motors are in this contracted state. Okay, so when this state is occurring, the myosin motor head has a low affinity for the ADP because it's its product. So it's going to release it into the cytoplasm. There's also just a much lower concentration of ADP compared to ATP in the cytoplasm. So just in terms of equilibrium, it's more favorable for the ADP to be unbound. And so it'll release from the myosin motor head. And when that's released from the myosin motor head, then it is free now to bind ATP. So when there's no more ADP, ATP can come in and bind, but then this creates now a weak association with actin. So when ADP was bound, has a strong association with actin. Now ATP is bound, and it has a weaker association. So now it's free floating. It's still closely like nearby the actin filament, but it's no longer bound to it. And because this myosin motor head is an enzyme, after ATP binds, it's going to do its enzymatic activity. So it's going to cleave this third phosphate and create an ADP and organic phosphate bound in the active site. And so this is really important because this cleavage, this enzymatic activity, drastically changes the conformation of this myosin motor head. So you can see in this step here, it's still perpendicular, but as soon as this enzymatic activity occurs, it kind of pulls back or stretches out this protein, kind of cocks it back. I guess this is like a gun analogy or something. And it's still unbound to the actin. But as we were saying before, when ADP is bound, this has a stronger affinity for the actin filament. So once this enzymatic activity occurs, then the myosin will rebind to actin. And so that's this step right here. So now the myosin motor head is bound to actin, but the enzymatic activity has occurred, so it's ADP and phosphate, and it has a low affinity for that phosphate. It's going to release into the cytoplasm, and this step is irreversible. And so similar to how when the catalysis occurred, or the enzymatic activity occurred, I guess it is a catalyst, so you could talk about it as catalysis. When this ATP is cleaved into ADP, that changed the conformation. Similar when the phosphate is released, that also changes the conformation back. And so this step is where the force generation occurs. It's rebound to actin, and the change in the conformation is going to pull that actin filament and collectively drive this contraction of these sarcomeres. And so then we're left back with the ADP where we started. Is there any questions about that? I tried to walk through slowly. I know there's kind of a lot of steps and things going on, but helpful to think through it. This is a really important concept. How about one question? I know it's late in the day. It's easy to be sleepy, but. All right. Well, one thing I want to point out here is some of these steps are in equilibrium, which means they can go in the forward or reverse direction. So this binding and unbinding of ADP is one of those. And this ATP to ADP step is also reversible. So this is an enzyme. A lot of enzymes can do this, but especially because the phosphate is still bound to the enzyme, it's still in close proximity. So this molecule can go in between ATP and ADP. So this is reversible as well as the binding and unbinding step. But once the phosphate is released, this is not a reversible step. So this step is kind of what is driving this cycle forward continuously. Yes. Yeah, so that's a great question. It is dependent on the availability of these binding sites. Yes. So yeah, strong and weak refers to the affinity of myosin to the actin filament. So the thin to the thick filament at these cross-bridge sites. Thanks for the questions. Yeah. So this is for striated muscle. So these sarcomeres specifically. We'll talk about smooth muscle on the next slide. Great. Okay, so smooth muscle is regulated in a slightly different way, and this is more representative of how most types of myosin are regulated. And so, like I said, we have these actin-based regulation and these myosin-based regulation, and so this is going to be governed by the myosin-based regulations. So this is governed by the regulatory light chain. That's what we were talking about earlier, and we'll come back to this in a little more detail. But interestingly, this is also triggered by the release of calcium. So the same upstream signaling event that releases calcium into the cytoplasm, granted these will be in different cells, but the same upstream mechanism will trigger the contraction of both smooth and striated muscle. So what this looks like in smooth muscle is more so this mesh network of different thin and thick filament associations that kind of condense the muscle cell or release it. So it's the same kind of overlap of these thin and thick filaments driven by the myosins moving across the filaments, but what the organization of it looks like is different compared to the sarcomeres in the striated muscle. Okay. So this regulatory light chain, you can think of it, oh yes. So I know it's really small, but the thick filament is here in the middle and the thin filament is here on the outside. So as long as the positive end is in this direction, these myosin filaments can move in that direction. And the same thing with this thick filament down here and this thick filament down here. And that'll lead to the condensing of these filaments over each other. Yeah, good question. Okay, so the regulatory light chain, we mentioned this a little bit earlier. This is kind of, you can think of this as like an accessory protein on the myosin filaments. So they're connected at this hinge region. Where's my pointer? At this hinge region, this RLC, regulatory light chain. So in terms of what this looks like for movement, it would be right here at this hinge location. And so this is in smooth muscle. Like I said, it's triggered by the phosphorylation of the regulatory light chain. And this is specifically at this threonine at site 18 and serine at site 19. And so the phosphorylation at these two spots is going to release the regulatory light chain to allow this myosin motor head to move. So it's kind of, again, a disinhibition in terms of when it's phosphorylated, it allows movement and activity of these myosin motor heads. So hopefully some of these words are familiar, but this phosphorylation event is driven by a kinase. And so this is quite literally called the myosin light chain kinase. It's acting on these two locations. And so this calcium release upstream is going to activate this, this myosin light chain kinase to then start adding these phosphate groups at this specific location. And it's going to allow the activity of this myosin motor domain to act. So as soon as this calcium event stops and there's no longer an abundance of cytoplasmic calcium available, then this kinase will not be active and the phosphatases in the cytoplasm will come in and start removing these phosphate groups. So kinases add phosphate groups, phosphatases remove phosphate groups. And these two work counter to each other in this regulation. But the kinase in this example is activated by calcium. The phosphatase is kind of on at a base level the whole time, but only when the kinase activity stops does the phosphatase activity overtake it. Any questions about this? Yeah. That's a good question. I would think that both, because yeah, your calcium is going to be depleted. I traditionally think of it as ATP because there's other biological activity that goes on. Do you know, Najwa?
Would think that maybe ATP would be the limiting factor, but I think that you're spot on. Everything.
Yeah, I think because there's so many components in the cells that are using ATP, and the production of ATP would also have to be actively going, that would deplete first. But yeah, I think exactly like you said, that the calcium is also going to be depleted. Good question. Yeah. Yeah, this is not what is, yeah, this is not present on the muscle myosin. Yeah.
So this is like a pretend like quiz question, like off top of it down. So you were mentioning earlier that like calcium is an important part of that kinase, myosin light chain kinase. What type of so where to put this? What type of regulation on that kinase might promote the activity? What is the calcium doing to this kinase to promote its activity? Yeah. Yes.
Nice.
Exactly right. So this is kind of like piecing together like a couple different concepts that we learned. And so that the calcium is a divalent cation. It has a charge, and so the charge that it brings with it when it comes over to that kinase acts in an allosteric regulation to change the active site so that then it can catalyze the reaction by adding a phosphate group. See, all of these things connect.
Yeah, that's a good point. I think in terms of ways to study, thinking about how you can draw on concepts from other lectures into different ones is a good practice for the exam. Thank you for adding that point. That's good. All right, so again, kind of similar to how kinesins undergo a conformational change when they bind their cargo, we can think of the activation of smooth muscle myosins in a similar way, in terms of this phosphorylation event, then changes the conformation and makes these motor head domains available for activity. Okay, we're going to talk a little bit about the myosin cargo transport now. So we mentioned some of these myosins are involved in cell movement and contractility, but also many of them are involved in cargo transport similar to dyneins and kinesins. So this schematic kind of demonstrates well the distribution of these cytoskeletal filaments in the cell. So the green here are the microtubules that are coming out from the microtubule stabilizing center or organizing center. And these red lines on the outside are the actin filaments. So they form kind of this cortex on the edge of the cell. And like we talked about before, this can be involved with cell movement in terms of their polymerization. And so in order for cargo to be transported all the way to the edge of the cell, they'll eventually have to switch to actin and utilize these myosin motors in that case. So this is kind of like going the last mile, or I guess the first mile if they're coming into the cell. And so we can talk about these directions similarly to how we talked about tracing in the last lecture, where we have anterograde moving away from the nucleus and retrograde moving towards the nucleus. And so in this case, we'd have in the anterograde, we have transport from the microtubules to this actin cortex, or vice versa in the retrograde. So some of these different motors, we have different types of myosin that will go in both the plus and the minus end direction. So like this myosin 5, you don't have to remember these specific myosin names in terms of their direction, but of course the dyneins and kinesins you should know in terms of the dyneins going in the negative direction, kinesins going in the positive direction. So the coordination between all of these will enable the efficient transport of these cargoes to anywhere it needs to be in the cell. No, yeah, good question. The cortex is this actin filament network that is localized around the outside of the cell, or the periphery. Yes. So if they're diffusing to the outside of the cell, maybe they'll get caught up in a vesicle. Yeah, I think that's the general term, but we'll actually talk about on the next slide in terms of when these tracks overlap, how do they transfer? So for these cargoes to move between the actin and the microtubule filaments, there has to be some kind of handoff or track switching going on. And so some cool research that has shown how this happens is kind of in this tug of war type of scenario, where the force of either, in this case, the dyneins or the myosins, whichever one is stronger, will take over the cargo. So in this case, we have a bunch of dyneins moving along this microtubule. And because there's more, they have more force. And so this cargo is more likely to continue along the microtubule than switch onto the myosin on the actin filament. Where if there's about equal amount of force, then it's equally as likely to go either along the myosin or the remain on the dyneins. And then similarly, if there's more myosin available to transport, then it'll switch in that direction. So this paper is actually a really cool demonstration that kind of shows some of the optical trap technology that was mentioned some in the pre-lecture. So that's actually what they use to determine this theory here. All right, any questions about this? Yeah. Yeah, that's a good question. All right, I'll walk through the numbers a little bit. So this formula in the cargo is referring to the ratio of forces between either the myosin, so it's myosin 5 in this case, or dynein. So in this case, if the ratio of myosin 5 over dynein is below 1, that means that the dyneins have a greater force. And so this 85% down here is saying it's at least 85% likely to continue down the microtubule because there's less force on the myosin compared to the dynein. In this middle graph, it's giving this ratio, but in this scenario, the ratio is 1, which means there's an equal amount of force that the myosin motors are exerting compared to the dyneins. So in this case, it's an equal chance that it will either continue along actin, continue along the microtubule, or stop. Yes. That's a good question. I don't actually know. I would assume it's related to the transcriptional activity of the cell. So in terms of what signals the cell is receiving, it'll activate different parts of the genome and transcribe more of these dyneins or myosin proteins. But in terms of their actual location, maybe it's just in terms of the availability, where they happen to be. I imagine there's some kind of randomness to it, but eventually things will get to where they need to go. Yeah, that's a good question, an interesting one to think about. Yeah, so this third one is similar in terms of the ratio where myosin force is higher, so there's a greater than 95%. It'll go along the actin. Yes. Yeah. Yeah, so this is in terms of the entire transport of the cargo. This cortex, since it's only it's localized to the outside of the cell, then that's the last step in the transport before this cargo would exit the cell. Yeah. Are myosins the only, what was that? Yeah, the molecular motors. So yeah, the myosin family of molecular motors are the only ones that are associating with actin. So because this cortex is made up of actin filaments, it's only going to be different types of myosins that are regulating the transport, but there are different types of myosins. And so those different types will move in either direction and they'll bind different types of cargo. And so between the whole family of myosins, that could cover the transport at this membrane. Good question. All right, any other questions about this before we move on? Okay, so yeah, I recommend you check out this paper if you want to see another example of this optical trap and how it's used to determine something useful about this biology. All right, so what happens in heart failure? So we talked about some of the underlying conditions that can lead to heart failure, but in terms of molecular mechanisms and etiology, there's a lot of things that are going on, and some of these things will be different in terms of what the underlying condition is. So one of them, specifically with MI, this might be a loss of cardiomyocytes, so the death of the muscle cells, the striated muscle cells in the heart. So that can lead to a decrease in the activity of the heart. This can be changes in tissue integrity, so either from congenital heart disease or like a tearing of the tissue for some reason. A reduced availability of ATP. So like we talked about, ATP is driving this cross-bridge cycle, and if there's some kind of malfunction in the ATP enzyme that makes it, or other things, this would lead to a decrease in contractility. Because calcium is regulating the availability from the actin side in terms of the heart, this loss of calcium will also impair the ability for contractility in the heart, or any kind of protein abnormality in something that makes up the sarcomere. So we're going to talk now about this drug, omecamtiv mecarbil. I don't think I said that right at all. I'm so bad at pronouncing things. So this is what this protein looks like, and this is a drug that was developed and that is intended to kind of get at some of these different causes of heart failure, specifically regarding this cross-bridge cycle. So what this drug does is it increases the rate of force generation by altering the myosin-actin binding kinetics. So this is, again, part of this cross-bridge cycle we'll talk about specifically where, so it's binding myosin, but it's not binding at the ATP active site. So where it's binding and cleaving ATP, it's binding allosterically to myosin. And this binding induces a conformational change that increases its affinity. So it kind of helps speed up this cycle by increasing the affinity to for ATP. Specifically in terms of if we think about enzyme kinetics, it is decreasing the energy barrier between the weakly bound and strongly bound states of ATP to ADP. So in this case, it's this step in particular in which this phosphorylation event occurs. So as I was mentioning, these states can be in equilibrium, meaning they can go either in the forward or the reverse direction. But what this allosteric regulation does is it lowers the energy barrier to the progression in the forward direction so that it's more favorable to continue in the positive direction of this cycle, which leads to a greater activity of this cross-bridge cycle and more contraction of these myosin motors and sarcomeres. Yes.
And when cardiac myosin binding allosterically, does that like in addition to normal process where myosin binds
Yeah, good question. It's in addition. So all of these listed binding activities would still be occurring. And this is binding allosterically at a different site, different from where calcium might be binding, which is in the troponin complex in this case, different from this specific cross bridge where the thin and thick filaments bind. It's binding actin and changing the enzymatic activity there. So this is just some, oh, is it gonna play? Okay. So this is some of the data that they got from the preclinical trial, looking at the efficacy of this drug. So this is an MRI video of a dog heart that's contracting either without or with this drug. And so something interesting that they saw was that the drug in these preclinical models was able to increase the contractility or the force generated with each contraction while keeping the heart rate the same. So if we know that increased heart rate might put great damage on the heart over time. So they want to restore the function of the heart without raising the heart rate too much. And so, yes.
Is the utility of this drug limited to only certain etiologies of heart failure?
So they focus on types of heart failure specifically with reduced ejection fractions. So these are the types of heart failure that don't produce as much force in the heart to pump blood. So the clinical trial for this drug was actually run over the past year, and they unfortunately announced that it would not be approved. So some of this can help elucidate how difficult it is. You can see this paper with this preclinical data came out over a decade ago. So how long it takes for these drugs to go through clinical trials, number of barriers. In this case, the extent of efficacy of this drug in actual human trials was not convincing enough in terms of having enough of an influence to be approved by the FDA. So this company is still working on other drugs, and there's still many opportunities for better therapies for people with heart failure. So if this is something that you're interested in, this is actually a really big strength of the BME department here at UVA. The Cardiovascular Research Center is here at UVA, a long-standing center of tons of people doing cardiovascular research, and researchers coming at this from all different angles. So Jeff Sosserman works on the computational biology side, working on systems biology of the heart and heart failure, specifically a lot to do with myocardial infarction. Brent French works on genetic engineering approaches using these viral vectors to kind of reprogram the heart for different therapeutic purposes. Fred Epstein, he is no longer the chair of BME, I should update that. But his lab is still doing really cool work. He actually got promoted to the school level. So he's just climbing up. We have a great chair now, Shane Pierce-Cotler, but his lab's doing really cool work on the MRI space, looking into some of the metrics that you can get from the heart and how that can inform different therapeutic approaches. All right, so if there's no questions, feel free to come up after class if you want to talk about your hypothesis, and definitely make sure to come to office hours.